My name is Jean Loring. I'm from the Scripps Research Institute. I study pluripotent stem cells and their use for a variety of applications, including neurological disease and rescuing endangered species. Would you bring up, the first cell? So I want to start out with some really basic things. This is the Wikipedia definition of a stem cell. I changed it just a little bit, but I think you all know a stem cell is a cell that can regenerate itself and it can differentiate into more than one cell type. Next slide. I'll get it. Or I won't. Can you advance the slide? I'm trying to. Oh, there it is, sorry. Okay, that always happens. Okay, so I just wanted to point out that if a cell is only capable of making one cell type, even if it's undifferentiated, it's called a precursor. It's not a stem cell. There's an absolute requirement for multiple abilities to differentiate into different cell types, okay? So once we have our definition straight, I want to remind you that there are two very different types of stem cells. It's unfortunate they're both called stem cells. The seemingly adult mesenchymal stem cells, or adipose tissue stem cells, are capable of making only a couple of cell types, and they're all connective tissue. They can't make neurons. So I depict these as the straightforward Swiss Army knife. Pluripotent stem cells, however, can make every cell type in the body. So if we remember these two things, when you talk about stem cell, you're not necessarily talking about the same kind of stem cell. They're not all alike. And this represents the ability of pluripotent stem cells to give rise to every cell type in the body. On some of those edges, you can see some really valuable tissues that can be used for cell therapy or for screening for drugs. There are two sources of human pluripotent stem cells. There are embryonic stem cells. These were first made in 1998, first published in 1998. And these were made based on a method that was used to derive mouse embryonic stem cells in 1981. So the pluripotent stem cell field goes back a very long time. And the human cells are a bit different, but they have the same range of capabilities. They're harder to grow, they grow slower, et cetera. But otherwise, we're all working in the same area. The other method, which is increasingly popular because of the ability to make autologous cells for transplantation and for making disease-specific cell lines to study disease processes, development of disease process, are induced pluripotent stem cells. As I'm sure you know, they're made usually from skin biopsies or other tissues from volunteers. These fibroblasts are grown and can be cryopreserved. They can be then thawed out and then reprogrammed using a very straightforward method developed by Yamanaka. The method that we use is Sendai virus because we found that it has the advantage of being non-integrative and also is relatively efficient. But all of the reprogramming methods do not induce mutations. This is something that we put a lot of work into to demonstrate earlier this year. So reprogramming is benign. And the cells that you get at the end are, with some variation in their epigenetics, identical to embryonic stem cells. They have the same capabilities, they have the same gene expression signature, they have the same DNA methylation signature within the limits, within the variations that occur within each of those cell types. So with that as clarity, the two speakers we have today are going to talk about some really wide-ranging and extremely interesting new research. First, we'll have Gary Smith from the University of Michigan, who is going to talk about, in general, how you can use pluripotency as opposed to using adult stem cells for medicine and for clinical research. And then Uta Hovata, and I've known her for like 20 years, but I still don't know how to pronounce her name. Sorry, Uta. From the Karolinska, we'll talk about the challenges, which is probably the cutting edge issue right now, are the challenges of translating these cells into clinical use. So please welcome Gary from the University of Michigan.
Great. Thank you, Janine, for the introduction. Thank you all for coming to this session. As I begin, as was indicated, we'll talk about pluripotency and its use in medical research as well as understanding early development. And specifically, we will be talking almost all about human embryonic stem cells. The group I work with, I want to acknowledge before I get started. It's really their hard work, their thoughts, their ideas, their clarity, and working as a team that really helps us make the progress that we make. So a little bit of how these stem cells can be used, or these pluripotent cells can be used. One, what everybody really talks about is they're being used for regenerative medicine, or being able to take a pluripotent stem cell, derive it to a specialized cell, and then using that cell to either replace bone marrow, nerve cells, heart cells, or pancreatic cells like you might see in a need for in diabetes. That's really not what I will talk about today. Today, what I will talk about is the other side of use of pluripotent cells, and that is in modeling diseases. And so if you think about different diseases, we may know that a genetic defect causes a disease, but there's a lot of things that we don't know that happen in between. And it's really that knowledge of what happens in between that gives us the power to be able to move the science forward. So really looking at using pluripotent stem cells as disease models. One, to understand how the disease forms and progresses, and then secondly, to be able to start thinking about using those phenotypes of the disease in a dish to begin to start looking at therapeutic treatments. So this is just kind of a emphasis of what Janine said. Stem cells are not created equal, just like animals are not created equal. They're all animals there on this picture, but they are not equal, and the same is true with embryonic stem cells. And I would say that not necessarily equal to pluripotent stem cells in some instances, and again, it all depends on what you want to use the cells for. And again, very clearly not the same as adult stem cells. So, as I began my career in science as a researcher, I also wanted to be able to work in a field that affected people's lives. And that's what really drove me to be an embryologist and to work in the field of human-assisted reproductive technologies. And I did that for many years, and then began in the late 90s, early 2000s to work on stem cells. Why do we study human embryonic stem cells? One is they are the gold standard, inherent in what they do. They do become every cell of the human body. Pluripotent stem cells can also, induced pluripotent stem cells can. But there have been publications to demonstrate that they do have some epigenetic or developmental memory. And some of the studies we're doing right now where we're able to actually look at genetically linked iPS cells and human ES cells in the same diseases, we're seeing the same thing, is that there's differences in the states of methylation and some of the gene expression. Maybe not as a pluripotent stem cell, but definitely as they begin to differentiate into cell lineages. There's a resource issue here. I think a lot of people stay away from human embryonic stem cells because of the controversy. But I think a lot of people don't recognize where these embryos come from, and I'll demonstrate that to you. That there are really over 200 single gene defect diseases that are tested for in human embryos. When that happens, those human embryos that are affected in the past have always been discarded. And we at the University of Michigan have made an opportunity for couples to donate those tested and affected embryos to try to develop disease models to study these different monogenic diseases. And the last thing I would say is that and it comes back to that resource issue, is that, this is something that we can make a difference. We can actually use these embryos that are being discarded to make stem cells, distribute them to many different people that are doing research in the field, and actually try to understand these diseases. So human embryonic stem cells obviously come from the human embryo, the inner cell mass at about day 5, day 6. We isolate those cells. Those embryos themselves come from 2 sources. One, they come from couples who are doing in vitro fertilization for infertility. They make embryos. They usually make more than 5, 10 embryos. They can use those embryos to build their family, and many times they have embryos that remain after they've built their family that have been frozen. They are given the opportunity to donate those to other people or to discard those. It's their choice, or they can donate those to research. The group that most people don't think about are those couples that don't have infertility and they're doing in vitro fertilization because they know they've already had a child affected with a disease, a monogenic disease, or they know they carry it in their family, something like cystic fibrosis, Tay-Sachs, Duchenne's muscular dystrophy. These individuals will do in vitro fertilization so they can generate embryos in the laboratory. Single cell or couple cells can be taken from every embryo, and then every embryo can be determined to either be affected or not affected by the mutation that causes the disease. Those that are affected in the past have always been discarded. Now they can be donated to our research program to make these disease-specific embryonic stem cell lines. This just emphasizes the issue of genetic analysis of embryos. It can be done either on day 3 or on day 5, where just a few cells are taken from the embryo. It doesn't harm the embryo. The embryos can then be frozen, the genetics of each embryo assessed, and there are over 200, and that is continually increasing, number of diseases that are tested for in human embryos by this procedure called pre-implantation genetic diagnosis. So when we think about human ES cell modeling of single gene disorders, it really is this issue of we know a genetic problem causes a disease in a tissue, an organ, or in some cells. But how it actually starts and how it progresses are things we don't understand. And so that really represents a black box. That's something like a human embryonic stem cell that has a genetic mutation that we can then study as it develops into, let's say, a nerve. How that nerve then functions abnormally, we can begin to understand the disease itself. And again, all of this can be done in a petri dish. Things that we could never do in a human body, we can now start to do in a petri dish in a laboratory. The other thing I'll emphasize here is as we make these human embryonic stem cells, we are firm believers that we don't need to keep these. The best way to move forward to understanding these diseases is to get them out to the disease-specific researcher, people that know about Tay-Sachs, people that know about adrenoleukodystrophy. They're the ones that are going to be able to move the science forward, not my laboratory. So that's really comes to our goals: derive and distribute these disease-specific human embryonic stem cell models, as I'll show today, to the worldwide research community, to accelerate our understanding of what causes diseases and come up with new treatments or cures, and then to engage and educate the public. So here are a few of the different stem cell lines that we've made. And one of the things I'm going to point out here, these are all disease-specific. So these are all from embryos that were diagnosed with either Huntington's disease, aniridia, is a blindness, Charcot-Marie tooth disease, Marfan syndrome, or adrenoleukodystrophy. The other thing I want to point out here is what we're beginning to do is form groupings of these human embryonic stem cell lines. And some of them come from sibling embryos, as you see in the yellow, and others represented by the boxing in are for the same disease. This is going to be very important as we move forward because one stem cell line for one disease is not enough to study a disease, just like one person is not enough to study one disease. But we're now starting to become enabled with these genetically linked and genetically non-linked human embryonic stem cell lines with the same mutations that cause these diseases. So one of the things you can think about is this is going to enable us to begin to think about things like disease penetrance. Just because 2 or 3 siblings have the same genetic mutation doesn't mean they always have the same disease or the severity of the disease. But now we're beginning to be able to see that we have these model systems where we can study this with genetically linked disease-specific embryonic stem cell lines and non-genetically linked disease-specific stem cell lines. So what I'm going to do is work through a few of these that we've done and show you some of the results. As you make an embryonic stem cell line for a disease, it's really not that useful unless you can demonstrate that it has a phenotype or endpoint measure in a cell that's known to be affected by the disease. So that really is some of the first work that has to be done. So the first example I'm going to give to you is hypertrophic cardiomyopathy. This is a disease of the heart where it thickens, it becomes large, it beats abnormally, and the individual will die from cardiac arrest almost spontaneously. And you hear about this many times in young athletes, but it's not just in athletes that it happens, it can happen in anybody. So we've made a disease-specific human embryonic stem cell line for a protein called myosin-binding protein C3 that's mutated in a family. They donated the embryos that were affected and tested by PGD, or pre-implantation genetic diagnosis. We made the stem cell line, and then what we do is we take a control human embryonic stem cell line and the hypertrophic cardiomyopathy mutation human embryonic stem cell line, and over 30 days develop them into cardiomyocytes. And we get a very pure population of cardiomyocytes, and then we can study those, control as well as disease-specific. And so if you could please click on this slide for me. This just shows these cardiomyocytes when they came from normal human ES cell lines, or if they came from the hypertrophic cardiomyopathy mutation disease-specific human embryonic stem cell line. And you can see they beat quite differently. We've been able to quantify this and demonstrate that this is repeatable, and that these beating frequencies as well as arrhythmias exist in the disease-specific line, but not in the control line. We've also been able to demonstrate that the cardiomyocyte size is significantly larger in the cardiomyocytes coming from the disease-specific human ES cell line compared to controls, and that the structure that gives rise to the beating, the sarcomere, is in disarray, significantly more disarray in the disease-specific human embryonic stem cell line derived cardiomyocytes than those from the control. In this publication, we are also able to demonstrate that we could take and gene correct the human ES cell line as we make them into cardiomyocytes using an adenovirus with the correct insertion of myosin-binding protein C3. When we did this, as we developed the stem cells into cardiomyocytes, we found that the cardiomyocytes now were significantly smaller with gene correction, and that their sarcomere disarray was corrected and they began to beat normally. So this is a way in which in a Petri dish we can begin to think about how this disease forms and how we can actually prevent it from having a phenotype that copies what is seen in the body. Another example I want to give is adrenoleukodystrophy, or ALD. This is a disease that affects boys and young men. Most boys that have ALD do not survive beyond the age of 21 years of age. And I was fortunate to meet the president of the ALD Foundation there, David Cry, and he actually was able to, through his foundation, talk with a couple who was in the state of New Jersey who had already had a child affected with ALD that were planning on doing in vitro fertilization with PGD to have a child without ALD. And they wanted to know if they could donate their embryos to something that would do good if they had affected embryos. And so they donated them to us. They had their boy there, Gus, who actually is free of the mutation, ABCD1 mutation, and will not or should not have ALD. And then they donated those embryos that were tested and affected. We make human embryonic stem cell lines. And one of the problems in this disease is there's a problem in fatty acid metabolism. So now what we're doing together with John Fink at the University of Michigan and Ann Moser at Johns Hopkins is actually looking at the fatty acid metabolism of these human embryonic stem cells, again, controls, as well as those that have the ABCD1 mutation, as they develop into neurons. So side by side, we can sample cells as they develop into neural rosettes, into precursors, into cortical neurons, or into oligodendrocytes, and actually look at how these cells are metabolizing fatty acids, which is one of the problems in the cell that's affected. The last example I'll give here is a neat one also, I think. This one's a little earlier in study, but this is for a disease called spinal cerebellar ataxia 3. This is a neurodegenerative disease. We were able to get embryos that were tested and affected with this disease, or at least had the mutation for spinal cerebellar ataxia C3. Working together with Hank Paulson and a few of his graduate students, we've been able to convert these into neurons and then look at the cellular localization of a protein called ataxin 3. Ataxin 3 is known to be abnormally located in nerve cells of patients that are affected with SCA3. And in fact, it's supposed to be a nuclear protein. And I don't know if you can see it because of lights, but we've been able to demonstrate that in these cortical neurons, ataxin T3 from the affected or disease-specific embryo or embryonic stem cell line actually is cytoplasmic, not nuclear, just like you would see in somebody's nerves that's affected by SCA3. So the beauty of this is that now we have an endpoint measure to say, yes, in the Petri dish, this disease phenocopies some of the characteristics of the nerves that are from somebody with the disease. So now we can begin to look at things like treating with small molecules, different genetic mechanisms or molecular mechanisms to be able to see if we can block this abnormal location of ataxin 3 in these cells as they develop in a Petri dish. The last thing I'm going to mention, and I showed that we have made recently a stem cell line for ALS or frontotemporal dementia. This is a line that had a nucleotide repeat in what's called C9, chromosome 9, open reading frame 72. And this is for a family that has affected people in their family line with frontotemporal dementia. So what we're looking at here is actually being able to make what we call multi-layered disease-specific, genetically linked stem cell models for specific diseases. And so we've done this for this ORF9 or C9 ORF72. And the example here is you might have an affected parent or carrier, an affected child. You perform PGD for the family to select embryos for an unaffected child, and they donate the PGD tested and affected embryos to us to make the disease-specific embryonic stem cell line. But at the same time, we can take biopsies, get skin cells from the carrier or affected parent or grandparent. We can also get it from a sibling. We can also get it from an unaffected parent or grandparent and make control-induced pluripotent stem cell lines. So now you have a grouping of pluripotent lines that you can use to study this disease in one family, in a second family, a third family, and more. So it will take time to develop these models, but I do believe that outside of the human body, these are really our best methods to be able to move forward in understanding many of the diseases that we can't actually understand within the body. So if we think about human embryonic stem cells that are disease-specific, they actually are an enabling resource. that allow us to study things that, like our neurodegenerative diseases, might be interesting to neuroscientists, cardiovascular diseases, pediatric diseases, diseases affecting vision, things that are studied in medicine, like sickle cell anemia, and also some cancers like retinoblastoma. As I end, I want to acknowledge the funders for the research that we've done over the last few years. In 2009, what was called Prop 2, the amendment to our state constitution at the state of Michigan, was passed, allowing stem cell derivation. Since that time, Mr. Alfred Taubman, the presidents of the university, the executive vice presidents of the medical center, deans, and other individuals have been incredibly supportive of this project. And lastly, if you know or you think you know of anybody that might be thinking about doing in vitro fertilization with PGD, the donation of these are very simple. It's all informed consent. All of our embryonic stem cell lines we make have been approved and accepted on NIH registry, which means that all of the paperwork is reviewed for every stem cell line. And the neat thing there is once those lines are accepted on the registry, anybody can get those stem cell lines from the University of Michigan. And while we cannot use federal funds to make the stem cell lines, once they're on the registry, anybody using those stem cell lines can use federal funds to work on them. So thank you very much.
We'll take questions at the end in our discussion. The next speaker is Uta, who will be talking about trying to take these sorts of therapies into clinical trials and some of the challenges.
Yeah, good morning. I'm very grateful for this invitation to this excellent meeting here. This little kitten is lynx lynx, the same species as you call bobcat in California. Not quite same, but relative. It's very rare to see them in the wood, particular kittens in pines. We are lucky now and then, and this one was in my own wood close to Helsinki. They climb to trees when the mother lynx warns them by moo, and if you hear the voice, you have to start looking at the trees. We are doing very rare things also in science, and sometimes we are lucky. For the most time, it is very tedious, systematic work we are doing with these cells and cell lines which we happen to get. Referring to Gary's talk, we have also some rare diseases from which we have got disease-specific cell lines. One homozygous Huntington's disease line and one ALS, amyotrophic lateral sclerosis lines. They are available to researchers. The main challenges before we can start treating diseases by stem cell-derived cell transplantation are that they can be rejected and that they eventually may form tumors when we don't treat them correctly. We can't inject pluripotent cells because they would form like clumps of cells called teratomas. But when we differentiate, we have believed that they are quite safe, but that's not so easy. We have to be very careful to exclude remaining pluripotent cells and to find manners to avoid rejection of the cells, because they are always allogenic cells to the recipient. We have been studying for a long time tolerance induction, so we made the cells tolerable to the recipient. And a system we have been using is co-stimulation blockade. We have results from that already from 2007. Another manner would be to give reaction-preventing drugs, for instance cyclosporine, that is widely used today after stem cell transplantation. The problem has been that it is toxic. For instance, these individuals who have got retinal pigment epithelial cells to treatment of age-dependent macular degeneration, they have needed large doses of cyclosporine in order to keep the cells in their eyes. And this cyclosporine has caused kidney damages, which are revealed by hematuria, and that's not a good thing for the patient. It's also expensive. In the eye, it's possible to inject cyclosporine intraocularly to the eye. In our experiments in Stockholm, it has prevented reaction, but there are other diseases which can't be locally treated after transplantation. So this co-stimulation blockade works in a manner that it prevents the contact between antibody presenting cells, dendritic cells in this case, and T cells that produce the antibodies, regulatory T cells. So can use two antibodies that prevent this co-stimulation contact, which is necessary for human immune defense, and it appears to work now pretty well. We are further testing it in animal experiments. We just use two antibodies at the same time as we inject the cells, and it works relatively well. Here we have used it in intratesticular injection of embryonic stem cells or retinal pigment epithelial cells, and it has worked very well. This is from an experiment which we made in Nova Southeastern University in Florida together with my postdoc Benjamin Joshi. This is a subcutaneous injection of these differentiated RPE cells from our Stockholm cell line HS401, which is also listed in the NIH stem cell registry. So this can be used for NIH support, federal support here in the US. No, I don't get this pointer to work, but you see these brown spots there within the transplant. And they are RPE cells, and these cells have been there for 60 days. The substance around them is matrigel, just to keep them together. In NSG mice, which are immunocompetent, it's another way how we can test them, make them to stay, is to use immunocompetent animals. But human recipients are always immunocompetent, and this does not save them. You see in a little bit more detail how they... No, this pointer doesn't work. They form clumps of cells, which we can also see on the Bruch's membrane in the eyes of animals into which we have injected them. And here with the red arrow, you see CD8 positive stem cell increase after injection of these cells. this is after 30 days, but after 60 days, this had disappeared. We can also make cells that are tolerated by the recipient, but there we need gene modification. Other groups and we have been using CRISPR-Cas9 technique to remove the most immunogenic parts of the cells. It's not without possible effects either, because it's gene modification, but so far it appears to be safe. Depends, of course, on what we are removing or adding to the cells. Several teams are just removing HLA tissue type determining segments, but that's not sufficient to make them tolerable to the organism. So we are also removing some sites and using which we can prevent natural killer cell activity, so that natural killer cells don't immediately eat up these cells. This is at early stages. We have to do more work before we can be convinced about their safety. One option is to make a large stem cell bank, like other stem cell types, for instance, umbilical cord stem cells, hematopoietic stem cells. It has been calculated that we would need at least 150 different haplotypes to select the most optimal cell line for each recipient. But when the starting point is human embryos, it's very difficult to get so many embryos with different haplotypes. In our laboratory in Stockholm, we have now 60 human embryonic stem cell lines, all made for research purposes. But those haplotypes are those which we have in Stockholm. Many immigrants though, but it's a very small minority thinking of global population. And it is extremely expensive to make these cells in a good manufacturing practice quality laboratory with all testing to fulfill clinical transplantation requirements, which must be fulfilled before we put these cells to patients. We have now come to that conclusion in Stockholm that without any company who participates and starts making them, it's not feasible for an academic group. We have made the work, but now somebody else can take over. We can stay with, but we don't have the funding. We have a good manufacturing practice quality laboratory in the same building as our laboratories are, and it works well, makes clinical grade cells for other projects. And we started making our ESL lines in that particular laboratory last summer this year. We can transport the embryos that have been made in an accredited in vitro fertilization laboratory and donated for stem cell treatments of other couples by the patient's consent. We have completely clinically feasible cells because we made them completely without any animal substances, so-called xeno-free cells. And these protocols on which we don't need any human fibroblast feeder cells or any other cells or any mouse-derived or bovine-derived substances. We only use human recombinant proteins, which we identified by together with my collaborator, Carl Tryggvason. And that combination of a human recombinant laminin 521 and E-cathepsin, which increases the cell-to-cell contact, that was published in Nature Biotechnology. And these xeno-free protocols are also available in an article 2 years ago in Nature Protocols. We had to change the culture medium. We now use Milteny's IPS brew and the same laminin from a Stockholm company Biolamina, we don't add extra albumin to cause any possible side effects to the process. And these media are completely just how they look like on this laminin surface 7 days after isolation from the embryo. And this laminin, it's a prototype still. We use human recombinant trypsin in passaging them. Then after 10 passages, we can start characterization and storing the cells. For genotyping, we have been doing SNP arrays, single nucleotide polymorphism arrays, and then sequencing too. Karyotyping we do, but it's not, it doesn't show many enough details. So we first, we take samples for the first time already after the first colonies have been formed. But we made a pre-master bank from which we can take one colony for further characterization, also for teratoma arrays, because that is necessary to see if they are tumor-forming cells or not. Even though all these genomic tests are normal, we don't know if there is something that still forms tumors in the genome, some mutations that are unknown in that respect. And these teratoma assays, we just inject either differentiated or non-differentiated cells to immunocompetent mice and let them grow as a tumor there. And when we used this NGS mice, we unfortunately noticed that one of our best looking lines, nice cells, karyotypically normal, no major SNP changes, it forms infiltrating tumors in the mice. Goes very nice to retinal pigment epithelium and makes dopamine producing neurons. And now we can't use that line. We can't take the risk that it starts infiltrating in the recipient. This was a new concern. It was not new, but these experiments unfortunately showed that this teratoma testing, in spite of all biochemical and genetic tests, must be done before we can go to clinical trials. But that's something that we soon know. This work has been done in 2 universities, Karolinska Institute, in which I've been working since '98, and Nova Southeastern University in Davie, in Florida, where I now have affiliation and where we can do all these safety tests too. Thank you very much for your attention.
Do we need microphones to talk from over here? Yes? Should we get one of those? Oh, okay, we get a special microphone up here. I want to spend the last bit of time we have, about 10 minutes, in discussing some of these interesting results. I think that the 2 talks that we had were really dovetailed in a way that I think is unusual because there aren't that many researchers that are focusing on embryonic stem cells these days. It's become more and more popular as it's become easier and easier to make embryonic stem cell lines. We can share. I'm going to sit here too. Yeah.
It's not turned up yet. You have to give me a chance.
I don't think we're on. So does anyone in the audience have questions? Would you come up to the microphone if you do? Okay, so I'll ask all the questions. Okay, could you turn on these mics? Okay, so now I get to sit down. This is actually a better mic. I'm going to change for the next time. So I think the interesting thing that was most interesting thing to me is the issue of of developing cells that aren't rejected, having some clever way of doing that, or figuring out a way around it, or immunosuppressing. And then the issue of tumorigenicity. Uti, I was going to point out that what I saw in those pictures didn't look like a teratoma because it didn't have all the germ layers. At least I didn't see all the germ layers. So I was wondering if maybe once your cells start differentiating, they're still tumorigenic, but they don't really form all of the possible germ layers.
I didn't show any teratoma injections. Those cells which we injected, they were differentiated, and we injected them to show that they don't form teratomas. Right. They were RPE cells.
Right. So they don't form teratomas, but they still invade tissues, which is, I think, something we really have to consider moving forward in the clinical area.
This is a new, very severe concern.
Yeah. So the FDA does require very extensive safety tests. So presumably, in the rats we actually have right now, we'll find out whether our cells form tumors. But I think that this is an important issue because where a lot of the FDA regulations right now, or the regulations that are implied by what the FDA has said so far, really don't cover all these possibilities necessarily with the right emphasis. In other words, it's really important to do karyotyping, but as Uti said, it's also very important to look for copy number variations and to do genotyping. So the fact that you didn't see anything that was obviously awry in your cells that form tumors is, of course, a concern.
Yes, because we don't know now what we should look.
Right. So would you care to?
Yeah, I would agree that beyond just your basic karyotype and your karyotype over time, I mean, I think is another very important thing. If you're just doing karyotypes, one of the things we've seen in many of our human embryonic stem cell lines that we've made is genetic consistency. And I say that genetic consistency over 150 passages being 7 to 10 days. So we're talking about an extended period of time. This is one of the things I'm finding with some of the iPS cell lines, as well as many of the lines that many of our colleagues are making, is the genetic instability of those lines. And
so I can comment on that a bit more.
Yeah.
We've done some really long-term studies, and I really mean long-term 2-year studies, continuous growth of ES cell lines. And we started to see mutations. I mean, we've shown that there's accumulation of duplications, copy number variations that increase the viability of the cells. They either cause the cells to divide a little more quickly, or they make them more resistant to apoptosis, or when they get passage, they stick down better. So there's evolution in these cultures, and we have to accept it. We can't do anything about it. What we found after continuous growth for 2 years was we started seeing deletions of P53, which is an extremely concerning issue. I think, and we found them in independent cell lines. So I think one of if you really should not culture the cells for a really long time, and you should always check them. That's pretty much our mantra. The other thing is epigenetic instability. I'm not sure everyone knows that all these pluripotent stem cells, if they're female, the second X will often become activated, so you have two active X chromosomes. We don't know whether that's something to be concerned about or not. We also see that the cells lose imprinting. So these are things that we have to figure out whether they're important or not, and whether they might be responsible, for example, for some of these anomalies in which cells that are already differentiated continue to divide.
I think that's a really great feed point to the issue of our knowledge of stem cells is increasing, but it's not all-inclusive. And one of the things I think about, I saw the use of the ROCK inhibitor, and this is one of the things that we don't know what that necessarily ends up doing to. It may make it easier to thaw a cell and get them to seed and begin to grow, but whether or not that actually changes something, and many times you'll get stem cell lines, and sometimes they suggest you use a ROCK inhibitor, sometimes you don't use a ROCK inhibitor. And so I think we need to be a little careful in comparing things, because many times there are many variables that are feeding in that we don't even know about that actually can have biological influences. So I guess I'm not saying ROCK inhibitor does, but I do know that ROCK inhibitors have very sequential and important issues on many different cell types, because you're inhibiting many of the kinases and the phosphatases that regulate cell cycle.
These cells which I mentioned, which formed these infiltrating tumors, they had never seen a ROCK inhibitor. We don't need it in the derivation process, because this laminin 521 is the natural laminin which 3-day-old human embryo contains. It also solves the problem of several passages, because this natural matrix enables us to get 30 times more cells within the same time. We culture them, the passage is 4 to 5 days, and we get large numbers of cells on this very safe matrix, as we think. But still we saw this problem. These cells were not p53 deficient.
Oh, I know. I don't think anybody had ever seen that before. It was just extremely alarming to us. We wanted to find out what happened when we grew the cells under different conditions for long periods of time. It turns out that one condition under which we grew the cells, we did not get deletions of p53, and those were the ones that had been cultured the way that embryonic stem cells had first been cultured, and that is by plating them on mouse embryonic feeder layers and manually dissociating them. So you cut them up and transfer them. So if you do that for 2 years, your cells will probably be okay. However, I think what we need to keep in mind is that we can't predict. We have to just ask the cells what's going on. We can't predict what's happening with them. Even though we have an idea about something that should be good for them or something that should be bad for them, we have to prove that every single time. And there are methods for doing that. They've become more and more available. And I think what we really require is a larger scale cooperation in the field, so that we can agree on what standards we should be using. I realize it seems silly after all this time that we haven't actually done that, but we've made some inroads. When there was one example that we did early on, and I knew Uti will disagree with this, is we gathered hundreds of cell lines, embryonic stem cells and iPS cell lines from all over the world, and we did gene expression analysis on them so we could define the borders sort of a gene expression profile of pluripotent stem cells. And from that we developed an assay. It's a diagnostic assay called Pluritest, which is a non-animal, non-teratoma way of demonstrating your cells are pluripotent. Now it does, all the cells that we found that were pluripotent by our assay also formed teratomas. So we could distinguish whether the cells would form teratomas or not. However, we have not extended that into other cell types. We are doing that now. But it's a lot of work, and it requires, the only reason we could do it was because 50 people who were growing embryonic stem cell lines were willing to give them to us and let us test them with full knowledge that we might find something wrong with them. So that's the kind of cooperation we need in this field.
We did a wide collaboration where you were also involved in this International Stem Cell Initiative 3, and there our cells didn't stick out by any.
Yours are okay.
Yeah, they were mostly okay.
Yeah, so
But still, I'm puzzled because after doing all the necessary tests, these cells still formed these infiltrations, and that's why I am not so happy regarding my cell lines anymore.
I agree. Let me give. There's another example that is not yet published, but I heard about this work, and there's some issues with quality control of other cell lines, like fetal neural stem cells, that you have to acknowledge that they change in culture. And unfortunately, some colleagues of mine discovered that when they used cells that had been the same cell line but grown in 2 different ways in a mouse model of a disease, they found that in one case the cells cured the animals, and in the other case they actually formed these tumors in the animals and did not cure them. So these are allegedly the same cells acting very differently. So that's another one of those sort of things that we need to figure out. It's currently a mystery.
Well, and I think it's something that I touched on in the beginning, and I tell all of my students, is that today we say an embryonic stem cell or a pluripotent stem cell is normal based on 1, 2, 3, 4, 5 things. And we say that iPS cells and human ES cells are the same based on 1, 2, 3, 4, 5 things. But we're still early in the science, and next thing you know, when criteria 8, 9, 10, and 11 come up, we are going to be thinking that, well, maybe we didn't know everything, and we didn't, because it is a early area in the science. It's been around a long time, but actually the studying of human embryonic stem cells as well as iPS cells has really not been very extensive or inclusive.
In iPS cells, we know some mechanisms of deviation.
Go ahead. Oh, no, don't give that to me. There's a question from the audience, finally.
Hi, Uti. I had a question for Uti in particular about the xeno-free protocols, and I just wanted to ask you another question about those. Very impressive, and I wanted to just ask if you could talk a little bit more about the importance of the xeno-free. And I might have missed this, but do you have side-by-side tests of the xeno-free and non-xeno-free cultured cells and how they are different in an animal model?
We did those tests when we started this xeno-free cultures, and we didn't see any significant changes. But when they are xeno-free, we don't have the risk of these animal-related viruses or agents, prions.
Okay, thank you.
So I think we need to wrap up. Just on the xeno-free issue, you know that the FDA has approved embryonic stem cell lines for clinical use that are by no means xeno-free. So there are mechanisms to, if you do the right testing, that the cells can be approved. So even though I think it's important, I don't think it's important for us to start over. I think we need to evolve these methods over time and try to use the best ones. We have to stop now. I greatly appreciate your attention, and I think this was a more interesting discussion than I thought it would be. Thank you very much, both of you.